Have you ever wondered exactly how your brain physically orchestrates movement and learning and emotion, like all at once without just crossing the wires?
Yeah, it's a lot to juggle.
Right. I mean, you reach for a cup of coffee, you remember a funny joke, and you feel a sudden wave of joy all simultaneously. How does the brain not just completely short circuit?
Well, to understand that, you really have to look at a structure deep inside your head called the basal ganglia.
The basal ganglia.
Exactly. It is essentially the brain's ultimate master switchboard.
And when the switchboard breaks down, I mean, the consequences are profound. We aren't just talking about a minor glitch here.
No, not at all. We're talking about severe life-altering conditions, like Parkinson's disease, Huntington's, autism, and schizophrenia.
It's massive. And for decades, neuroscientists have been trying to map exactly how this switchboard is actually wired.
Which is what brings us to today. Because a groundbreaking 2026 paper from Nature Neuroscience just came out.
Yes, by Kwangson Wang, Hanqi Zhang, and their huge team at the Allen Institute. And it has finally given us the blueprint.
It really has.
So the mission of this deep dive today is to exhaustively decode their completely new master map of the mouse basal ganglia. We are going to explore every concept, every mechanism, and honestly, every tiny genetic detail they uncovered. Okay, let's unpack this.
Well, to really grasp the magnitude of this map, we have to start at the physical gateway of this switchboard.
Right, the lobby.
Exactly. The basal ganglia isn't just one lump of tissue, it's a collection of interconnected nuclei. And the main entry point, the grand lobby where all the messages from the rest of your brain first arrive, is called the catoputamen.
Or the CP, for short.
Right, the CP. You might also know it as the dorsal striatum. So the CP receives these massive inputs from the cortex and then routes them down the line.
But the CP isn't just like empty space, right? It's packed with millions of highly specialized operators.
Yes, and the main operators in that lobby are medium spiny neurons, the MSNs.
Okay, MSNs. And I know they've traditionally been divided into two main pathways, but it sounds like the Allen Institute found, well, a lot more nuance there.
Oh, far more nuance. I mean, they looked incredibly closely at the actual genetic recipes of these medium spiny neurons.
And they divided them into what they call transcriptomic classes.
Yeah, T-types. Historically, like you mentioned, we thought of two main classes. First, the MSNs expressing D1 dopamine receptors.
Right, which form the direct pathway.
Exactly. And second, the MSNs expressing D2 receptors, which form the indirect pathway. But the researchers isolated a third, highly unusual class.
Atypical MSNs.
Yes. Specifically, a group known by this long genetic signature, FTRD1 Samafa GABA.
Okay, let's translate that functional difference for a second. Because the old analogy of D1 being the gas pedal and D2 being the brakes, it always felt a bit too simplistic to me.
It is a bit basic. Yeah.
Right. So if we think about this like a massive audio mixing board in a recording studio, the D1 neurons are like the volume dials for action, right? They're pushing specific behaviors forward.
Spot on.
And the D2 neurons are the mute buttons, actively suppressing competing background noise so your intended action comes through clearly.
I like that analogy.
But how does this atypical class, fit into that mixing board? It seems like a weird experimental hybrid transmission system or something.
That audio mixing analogy is actually perfect here. Because the atypical MSNs are fascinating in how they break the traditional rules.
How so?
Well, they actively express the D1 volume dial receptors, but they simultaneously express a short variant of the D2 mute button receptor.
Wait, both at the same time?
Both at once. Mechanistically, this creates a biological hybrid. It suggests these specific neurons have a localized internal feedback loop.
Oh, wow.
Yeah. They might be able to initiate a signal while simultaneously setting their own threshold for when to shut that signal off.
So they don't have to route the signal all the way back through the cortex.
Exactly. It allows for incredibly rapid behavioral flexibility.
That is wild. They basically have their own internal volume limiters built right into the receptor profile.
Yeah. And what's fascinating here is that the complexity doesn't just stop at those three broad classes.
There's more.
A lot more. When the researchers zoomed in even further on the genetic transcriptomes, they discovered that within those three groups, the D1 dials, the D2 mute buttons, and the hybrids, there are actually 24 distinct transcriptomic subtypes operating inside the catoputamen.
Wait, 24 distinct T-types?
Yes. 24 unique genetic identities for these operators.
24 distinct operator types just in the lobby of the switchboard alone.
Just in the CP, yeah.
So before we move on, let's just recap that for a second. The basal ganglia controls movement and emotion. The CP is the gateway. And the MSNs, the D1, D2, and atypical neurons are the switches.
That's the core machinery.
So if you're listening to this, I mean, can you picture these millions of D1 and D2 neurons sitting in your head right now like tiny gas pedals and brakes?
It's a lot of traffic to manage.
It really is. Which brings up an immediate question. How does the brain actually keep them organized? And honestly, how do scientists even know there are 24 distinct MSNT types?
Right, the methodology.
Yeah, how do they know where they live without just like grinding the tissue down and losing the structural map entirely?
Well, that has always been the primary bottleneck in neuroscience. You either get the map or you get the genetics, but rarely both.
So how did they solve it?
They utilize massive data from the Allen Brain Cell Atlas, or ABC Atlas, and they combined two distinct, highly advanced technologies. First, they used cRNA-seq.
Single-cell RNA sequencing.
Right. You isolate individual cells and sequence their RNA to figure out their exact genetic recipe. It gives you the comprehensive parts list.
But like I said, sequencing alone destroys the tissue, right?
It does. You know exactly what the cells are, but you have no idea where they were originally located.
So you need a way to find their addresses.
Exactly. To solve the location problem, they deployed MERFISH.
Ah, MERFISH. Multiplexed error-robust fluorescence in situ hybridization. I've read a bit about this. It allows you to actually see the molecules inside intact tissue, doesn't it?
Essentially, yes. MERFISH allows scientists to take an intact three-dimensional slice of brain tissue and use these fluorescent probes to tag specific RNA molecules.
Directly inside the cells.
Directly inside the cells while they're still sitting in their natural physical architecture.
Oh, that's amazing.
So the cRNA-seq gives you the recipe, and MERFISH shows you exactly, physically, where those genetic recipes are located in the intact brain.
Okay, but even with that tech, you're generating an absolute ocean of data. I mean, if you have millions of data points glowing in a brain slice showing where 24 different types of neurons are living.
It's overwhelming.
Yeah. How do you mathematically make sense of that? I know the paper leans heavily on an algorithm called non-negative matrix factorization, NMF.
Yes, NMF is the key here.
But I really want to understand the actual mechanism of how that algorithm organizes biological chaos. Can you break that down?
The mechanism of NMF is actually incredibly elegant. Imagine taking the brain and dividing it up into a microscopic grid.
Okay, a grid.
And each little box, or bin in this grid, is just 100 micrometers wide.
Tiny. Super tiny.
NMF then calculates the spatial histograms, which is just the exact counts and proportions of different cell types within every single one of those tiny bins.
So it's counting who lives in each microscopic neighborhood.
Precisely. It is essentially looking for high-dimensional co-occurrence. NMF mathematically asks, out of these 24 cell types, which ones consistently show up together in the exact same 100 micrometer bins across the entire brain.
Interesting.
Right, and by factorizing that matrix, it distills millions of scattered data points into a dictionary of underlying spatial motifs.
Ah, I see. Okay, so if we think about this visually, imagine pouring a fully blended smoothie through a magic NMF filter.
A smoothie filter. Okay, I'm with you.
Yeah, and instead of just getting a finer liquid, this filter instantly separates the smoothie back out into a perfectly organized grid of whole strawberries, whole bananas, and individual spinach leaves.
That's a brilliant way to conceptualize it. It isolates the ingredients perfectly.
It unmixes the data.
Exactly. And when they ran the Kaduputaman data through this NMF algorithm, the fundamental finding was pretty astounding.
What did they find?
The algorithm revealed exactly 10 distinct spatial patterns of MSNs in the CP.
Just 10 patterns for all 24 types.
Yep. Furthermore, most of those 24 different genetic subtypes of neurons strongly belong to just one specific spatial pattern.
So they aren't just scattered randomly everywhere?
Not at all. They are highly organized into specific segregated neighborhoods based on their genetic barcodes.
Okay, let's do another quick recap. cRNA-seq finds the genetic recipe, MERFISH finds the location, and the NMF algorithm groups them into 10 spatial patterns.
That's the toolkit in a nutshell.
So ask yourself as you listen to this, if we looked at a 3D blueprint of your brain's genes right now, would it look like a random Jackson Pollock painting or a perfectly organized mosaic?
It's definitely a perfectly organized mosaic.
Right. But those genetic mosaics aren't just abstract data on a computer screen. They perfectly align with physical 3D anatomical structures in the brain.
They do.
So how did the researchers bridge that gap between the genetic map and the physical anatomy?
They registered all of these genetic patterns to the CCFV3.
Which is the Common Coordinate Framework version 3.
Exactly. Think of the CCFV3 as the definitive three-dimensional Google Earth base map for the mouse brain.
Okay, a 3D Google Earth for the brain.
Right. And by layering the NMF genetic data and the physical cell density onto this 3D framework, they discovered that the cataputamen is actually divided into 5 specific kingdoms.
5 kingdoms.
5 3D subdivisions. And each one has a unique cellular architecture, unique marker genes, and entirely unique functional roles.
5 distinct kingdoms inside the cataputamen. Okay, I actually have a mnemonic device for you to remember these 5 CP zones if you're trying to visualize this at home.
Oh, let's hear it.
Just remember, dark vampires love icy places.
Dark vampires love icy places.
Yep. That stands for dorsomedial, ventromedial, lateral, intermediate ventral, and posterior.
In fact, it's really helpful.
It makes it stick. So let's dive deep into the specific mechanics of each of these kingdoms, starting with the dark vampires, the dorsomedial and ventromedial zones. What makes them functionally distinct?
Well, let's look at the dorsomedial CP first, or CPDM. Under a microscope, this region physically exhibits these smaller, darker clusters of neurons.
Darker clusters, hence the dark vampires.
Right. And historically, we call these structures striosomes. But genetically, the CPDM is defined by the high expression of a specific gene called FOS.
FOS, F-O-S.
Yes. FOS is an immediate early gene, which is famously linked to rapid neuroplasticity and learning.
Oh, interesting.
And functionally, this makes perfect sense because the CPDM receives major inputs from the anterior cingulate area and the visual cortex.
So it's processing what you see and how you feel about it?
Exactly. It is constantly updating your behavior based on complex, dynamic, emotional, and visual environments.
So the FOS gene equips those specific neurons to adapt really quickly. That makes sense. What about the ventromedial CP?
The CPVM. It also contains those dark striosome clusters, but its defining marker gene is Col6A1.
Col6A1, what does that gene do?
It's heavily involved in the extracellular matrix.
Like the scaffolding between the cells.
Exactly. The structural scaffolding. This implies a very different physical and metabolic environment compared to the CPDM.
And what kind of inputs does it get?
Its inputs mirror this difference perfectly. The CPVM is targeted heavily by the prelimbic and orbital areas of the prefrontal cortex.
Okay, so prefrontal areas.
Right, which are regions deeply tied to expected rewards, motivation, and goal-directed behaviors.
Okay, so that covers the darker, more cognitive and emotional regions. Moving on in our mnemonic to love icy places, lateral, intermediate ventral, and posterior.
Let's start with the lateral CP, or CPL. Physically, it looks entirely different from the medial sections. It's a brighter, much denser structure on the imaging scans.
Brighter and denser.
And its defining marker gene is ASTAN2. Functionally, the CPL is your primary motor hub.
Just raw movement.
Raw movement. It is overwhelmingly targeted by somatosensory and motor inputs from the cortex. If you are physically executing a movement, like reaching for that coffee we mentioned earlier, the CPL is running the show.
Okay, and next to it is the intermediate ventral CP, the CPF.
Right, the icy in your mnemonic. It shares that bright, dense appearance with the lateral section, but its defining gene is WFS1.
Wait, WFS1, isn't that classically associated with cellular stress responses and calcium regulation?
It absolutely is. And again, the genetics match the function beautifully. The CPFAV receives inputs from the anterior, insular, gustatory, and visceral cortices.
Gustatory and visceral, so like taste and internal organ signals.
Exactly. It is the center for processing internal bodily states, autonomic stress, and taste. So having high calcium regulation makes total sense for a region dealing with continuous, slow-wave internal homeostatic signals.
That is so elegant. And finally, we have the smallest kingdom, the places, the posterior CP or CPP.
The CPP is characterized by the OTOF gene, and its wiring is highly singular.
Singular how?
It is driven almost entirely by auditory inputs from the cortex. It is essentially the specific terminal where the basal ganglia listens to the outside world.
So depending on what you were doing, totally different parts of this lobby light up. Which means, if you're listening right now, that OTOF gene in the CPP is doing the heavy lifting. It's literally routing my voice through your switchboard at this exact moment.
It really is.
And what blows my mind is the sheer scale of this project. Because they didn't just map the cataputamen, they applied this exact same 3D coordinate mapping to the entire basal ganglia pathway, didn't they?
Oh, they mapped the entire subcortical universe. They tracked these gene expressions downstream and found that the globus pallidus externa, or GPE, divides into medial and lateral zones.
Wow.
The globus pallidus interna, the GPI, split into a core and a shell. The subthalamic nucleus, or STN, divides into posteroventral and anterodorsal zones.
They just kept going.
They did. The substantia nigra pars reticulata, the SDNR, divides into 5 distinct subdivisions, and a crucial thalamic relay node called the parafascicular nucleus, or PF, splits into 4.
So they essentially established the definitive genetic borders for anatomical regions that neuroscientists have been arguing about for, what, half a century?
At least half a century.
It is a breathtaking amount of clarity. Let's do another quick summary. We have 5 CP zones: CPDM, CQVM, CPL, CPIS, and CPP, and each is defined by specific genetics and tasks.
Perfect summary.
So, listener, which of these kingdoms is working hardest right now as you listen to this deep dive? Hint, it's the auditory-driven CPP.
Definitely the CPP.
But,
Yeah. By using massive arrays of anterograde viruses, which travel forward down the axon to show where a signal goes, and retrograde viruses, which travel backward to show where a signal comes from, they literally mapped the entire cortical output.
So they can see the exact start and end points.
Exactly. And they found that the cortex is partitioned into 6 massive functional modules: prefrontal, lateral, somatomotor, medial, visual, and auditory.
6 cortical modules.
And these modules preferentially target the specific CP subdivisions we just talked about.
So the visual module targets the CPDM, the auditory targets the CPP, and so on.
You got it.
But let me challenge that for a second. If the somatomotor module in the cortex is just like blasting the lateral CP with a massive wave of activity, how do we not just flail our arms around constantly?
That's a great question.
Right, because how does the basal ganglia filter that signal so that I only move my index finger without accidentally triggering an emotional response or just moving my entire body at once? Surely not all neurons sending messages are doing the exact same job.
They definitely aren't. And that is the exact physiological puzzle here. It is solved by the specific dual wiring nature of the output neurons themselves.
Dual wiring.
Yeah. The researchers looked incredibly closely at layer 5 of the cortex, which is the main output layer sending signals down into the basal ganglia. And they found 2 totally distinct classes of messenger cells doing profoundly different jobs to prevent that exact flailing you described.
Okay, what's the first class?
First, you have the layer 5 IT neurons. IT stands for intratelencephalic. These neurons provide the strongest innervation to the CP.
So they do the heavy lifting for the signal.
Yes. Their terminal fields are broad, they're widespread, and critically, they often project bilaterally.
Bilaterally, as in to both sides.
Yes. A single layer 5 IT neuron sends its message to the cataputamen in both the left and right hemispheres of the brain simultaneously.
Here's where it gets really interesting. If we think about how a massive organization communicates, the layer 5 IT neuron is basically a company-wide reply-all email blast.
A reply all. I love that.
It's broadcasting this broad, powerful, bilateral message to both sides of the brain, ensuring that both hemispheres are continuously coordinated and aware of the general motor plan, right?
Precisely.
But to execute that highly specific finger movement, you need the other type.
Exactly. The other type is the layer 5 ET neuron. ET stands for extratelencephalic. And these neurons are the precise opposite of a reply all.
What do?
They produce incredibly narrow, highly focused topographic projections. They project strictly ipsilaterally, meaning they stay entirely on one side of the brain.
Only one side.
And most importantly, ET neurons don't just stop in the cataputamen. Their axons punch straight through the lobby and directly target the downstream executive nuclei, like the GPE, the STN, and the S-senior.
All right, so to stick with the corporate analogy, the layer 5 ET neurons are like highly secure, local direct messages sent to specific downstream executives.
Yes.
They completely bypass the broad company chatter to execute a precise, localized command.
If we connect this to the bigger picture, this specific dual wiring system is just a marvel of biological engineering.
It really is.
Because you have topographical, organization-specific functional modules in the cortex talking to specific genetic kingdoms in the CP. But you simultaneously have this overlapping modularity of reply-all IT neurons and direct message ET neurons.
Working at the same time.
Exactly. This combination is what allows your brain to process parallel streams of information like maintaining your bilateral posture while walking, while simultaneously integrating a precise, single-sided action, like reaching out to catch a ball with one hand.
It is how you act without short-circuiting.
Exactly.
Okay, let's bullet point this section. 6 cortical modules wire into the 5 CP zones. Layer 5 IT neurons send broad dual hemisphere signals. Layer 5 ET neurons send narrow single-sided signals.
Perfect.
So think about your own daily routines, listener, those deep-seated habits that just feel completely automatic. How does understanding this specific reply-all versus direct message wiring change how you view those behaviors?
It makes you realize it's not just some vague psychological loop.
Right. It is a physical, genetically defined pathway being carved into specific coordinates of your brain.
It transforms our entire conceptual framework of habit, behavior, and particularly neurological disease.
So what does this all mean? When we step back from the single-cell RNA sequencing and the NMF algorithms and the viral tracing, what is the ultimate takeaway from Wang, Zhang, and the Allen Institute team?
The ultimate takeaway is that combinatorial gene expression is the literal foundation of the brain's functional architecture.
The genetics drive the structure.
Absolutely. The specific genetic recipe of a neuron dictates exactly where it lives in 3D space, what spatial pattern it belongs to, and precisely who it talks to across the entire brain.
So the basal ganglia is not a homogeneous mush of cells at all.
No, it is a globally mapped network of parallel, modular, and cell-type specific subnetworks.
It's essentially a GPS coordinate inextricably linked to a genetic barcode. That level of deterministic organization is just incredible.
It is. And, it raises a final, frankly, mind-expanding question that isn't directly answered in the text, but builds perfectly upon all this data.
Ooh, what is it?
If individual neurons have such highly specific modular roles based on their exact 3D coordinates and their exact genetic signature, could the future of medicine involve therapies that target a single genetic coordinate?
Wait, you mean instead of taking a pill that floods the entire brain with dopamine, causing widespread side effects, you create a therapy targeted to just one intersection on this new map?
Exactly. Imagine a future treatment for a severe motor tic or a compulsive habit. Using this map, a therapy could be engineered to only target the D2 MSNs located exclusively within the Aston2-defined lateral CP spatial pattern.
Oh my God.
Right. It would fix the specific motor circuit without altering a single neuron related to your emotions, your memories, or your personality in the neighboring medial CP.
Because you have the exact address.
Yes. By giving us the exact genetic and spatial blueprints of these networks, this paper lays the absolute foundation for that kind of hyper-precision medicine.
That is an unbelievable possibility to think about, a true paradigm shift in how we might eventually heal the brain without changing the mind. We're going to leave you to mull over that incredible possibility on your own. Thank you for joining us as we explored the microscopic kingdoms of the basal ganglia. Until next time, keep learning, keep wondering, and keep diving deep.